Hello, folks. I just want to introduce Chapter 10 to you folks. It's a pretty light chapter. A lot of it goes hand in hand with lab, so it's one of our shorter ones. So I'm going to go ahead and just present the recording to you, and later this coming week after spring break, I'm hoping to have some time either after lecture or lab where I can field any questions or just kind of review over the material in case anybody had any questions and concerns about this chapter. But anyways, think of questions as we go. Okay, so Chapter 10 just kind of fills in the gaps on some topics you may have encountered in general biology regarding the identification and classifications of organisms. More specifically, we're going to look at these schemes as they apply to bacteria. So going back to the first chapter of the semester, we did briefly discuss the fact that all living organisms, including microorganisms, are sorted out into different groups based on characteristics like phenotypes, so physical characteristics, or behavior, or even these days genetics. Okay, but the thing is that understanding how to identify and classify organisms is a big area of biology. It is so large it gets its own specific science, taxonomy. Okay, so this chapter is about the basics of bacterial taxonomy. So the science behind how we identify, classify, and even technically name bacteria. So we're going to look at these three areas today. We're going to elaborate on them. So we're going to look at identification schemes of bacteria. Okay, so when you have an unknown organism, what are the methods to use to identify that unknown organism? And that's the first step in figuring out who that organism is related to. Okay, so what taxonomic groups does it belong into? And then we're going to look at classification schemes. Okay, so once you have a bunch of identified organisms, what is the best way to decide who is related to who? Should you go by behavior? Should you go by genetics? What sort of scheme are you going to use there? And then lastly, and very simply, we're going to talk about nomenclature. So how do we decide what names to give new organisms? So let's just get nomenclature out of the way, because honestly, there's not much to it that we care about. So we have a new organism, or we even have to rename organisms. Who gets to make those decisions, right? Who decided that we're called Escherichia coli or Staphylococcus aureus? Well, these days, when we have a new organism, generally the name is debated upon and naming is given according to the International Code for the Nomenclature of Prokaryotes. Basically, a bunch of experts in the field set the rules for deciding what names we're going to give out. Sometimes we still give out fun names or we name it after a scientist, but not always. And these rules change a lot over time. As we mentioned in lab with the bioesculin plates, the group D streptococci were renamed maybe 15 years ago into the Enterococci. Okay, so I remember learning them as Streptococcus when now we talk about them as Enterococcus. So, these schemes do change over time, but that's as far as we're going with nomenclature. Alright, so we're going to look a little bit at classification first, and then we'll come back to it again later on, but just to introduce you to how some of this works. So, classification is such a big area of taxonomy that it gets its own sub-discipline, phylogeny. Okay, so there are specialists in phylogenies, people who specialize in deciding how we relate all organisms. Okay, so broadly defined, it's the study of evolutionary relatedness. Okay, so during evolutionary time, we have all these organisms. How to decide who shares what ancestors? Okay, so how do we decide to relate them that way? So in the past, organisms were classified, and still a little bit to this day, based on phenotypic attributes. Okay, this bacteria looks and behaves like this bacteria, therefore they're related. Okay? We don't really do that much anymore. Using phenotype isn't super reliable. What we're leaning on more these days is relying on genotypic differences. So comparing DNA and RNA sequences of various organisms to get a better idea of how they're related. And that's really kind of shaken things up in the last 20 years. But here's the sad truth of phylogeny. Okay, there's tons of classification schemes. There's all sorts of different ways that we can figure out who is related to who. So there is no such thing as an official classification system to this day. Okay, there are all sorts of them constantly being debated over. Phylogeneticists often get into a lot of pissing contests. It's pretty funny if you ever go to one of their talks. The Q&A sessions are always very controversial. So there's a number of different preferred classification systems, but overall, the most widely preferred is the one we talked about in Chapter 1, the 3-domain system. Okay, so this is where all living things either end up in domain Archaea, domain Bacteria, or domain Eukarya. Okay, so just to show you how fluid classification schemes are and how much they've changed over not that much time, I just want to show you folks a little backstory here. So prior to the 3-domain system, I learned about the 5-kingdom system. Okay, so what we have here in this slide is a 5-kingdom system. This is what I learned in high school in the late 1990s, and this is what most kids learned about going into the very, very early 2000s before it's replaced by the 3-domain system. So, now we have 3 domains, but prior, we sorted out all living things into 5 kingdoms. Okay, we have the animals, so Animalia, the plants, Plantae, fungi, Protista, and whatever the hell Monera is. Okay, so at the time, bacteria, archaeans, they were one and the same. We did not know they were different. So you get lumped in this weird group called the Monerans. If you go back even further, it gets worse. Okay, so first classification system was set up by Carolus Linnaeus. Okay, just a few hundred years ago. He just decided that, we're exploring this planet. We've encountered all these new and crazy animals and plants. Maybe we should start cataloging them and classifying them. So, the original classification system developed by Linnaeus had all living things as animal, plants, and then technically mineral, but they leave that off. Alright, 1866, Haeckel decides that, we've got these microorganisms around now, okay, which Linnaeus didn't see. So they need their group. So we got animals, plants, and then we got the Protista. Okay, Protista are all the microorganisms. And something to think about, folks. Where are my mushrooms? Okay, mushrooms were considered plants for a long, long time. So fungi were plants. So we go to 1959. Okay, Whittaker finally comes up with the 5-kingdom system, folks. This is the late 1950s. That's not that long ago, where we thought that fungi were still plants. So, 59, we adopt Whittaker's 5-kingdom system. So now we got animals, plants, fungi, Protista, and now the bacteria, and technically archaeans, get their own group, the Monerans. So they're the prokaryotes. They're in their separate group. So, what happened after that? How do we get to the 3-domain system? Okay, that's where the story gets kind of interesting. We made a lot of important discoveries thanks to one individual in his research group. So, between the 70s and 1990s, a gentleman named Carl Woese changed the face of phylogenetics. So, Carl Woese is the person we thank for the 3-domain system, the currently most believed in classification system, or widely accepted. Okay, so what makes a three-domain system better than the other systems? So five-kingdom system was a classification system based on phenotype. Okay, we sorted out these organisms the way we did because they look the same, they behave the same. But Carl started looking at genetics. He wanted to compare DNA sequences, and that's because of an organism he was working on. Carl was working on a group of prokaryotes called methanogens. So, methanogens, as we understand now, are archaeans. But he did not know that in the 1980s. He thought they were really weird bacteria that did not behave like most bacteria. And he just couldn't figure out where to put them in the grand scheme of things. They looked like bacteria, they behaved somewhat like bacteria, but they're really, really kind of different. So he started digging into their DNA. And specifically, Carl was looking at their genes for rRNA. Okay, so they want to look at the genes for their ribosomes. And he chose that because those genes for rRNA see very little mutation. Okay, so if organisms are very closely related, their rRNA genes will be very similar in sequence. So, Carl sequenced their rRNA genes and started comparing it to other bacteria. And what did he find? Their rRNA genes were really, really different than bacteria. And in fact, the rRNA genes of methanogens were more closely similar to that of eukaryotes. So he thought that was really weird. He's like, okay, I've got this group of what I think is bacteria, but genetically they're more closely related to eukaryotes like humans. So what do? Well, he started looking for more of these oddball organisms and started cataloging all sorts of rRNA gene sequences from all sorts of organisms. And the more he did this, the more he discovered that there was more of a three-domain system going on. That you've got bacteria, you've got the eukarya, and then you get the archaeans that are somewhat in between. They look and behave like bacteria. They are prokaryotes, but genetically they are distinct. So, this led to some other revelations. Okay, everyone and their brother has sequenced an rRNA gene sequence when working with new organisms. We have massive catalogs of rRNA gene sequences. So this started setting up what we call the molecular record. It allows us to start predicting and working backwards through evolutionary time to determine where mutations arose in these rRNA gene sequences. And because of the molecular record, because we can plot these mutations backward through time, we can plot them so far back that, in theory, we believe that there was LUCA. Okay, so folks, if you're unfamiliar with LUCA, LUCA stands for the last universal common ancestor. In short, LUCA is the original cell, the organism that gave rise to all other organisms. So think of it this way. It is the organism with the first rRNA genes that got shared and mutated through evolutionary time. So, because of this, because we started comparing all organisms based on rRNA and not phenotype, we discovered that there was, again, 3 domains, not 5 kingdoms. But this is probably going to change over time. So, just to show you a broad picture of what Carl Woese was trying to paint here, here is a very basic phylogenetic tree showing the 3 domains and where some of these branch points have occurred in the molecular record, where some of these mutations have happened, some of the more major splits. So you can see at the bottom here, at the base of the tree, that it should be LUCA, the universal cell. Okay, at some point there's a big split where bacteria went their own way, and then some proto-version of archaeans and eukaryotes went their own way. And then you hit another split here where archaeans and eukaryotes go their own way. And then there's just more and more splits over time, creating greater diversity, more and more organisms. Alright, just a kind of nifty chart, just kind of hammering home the point that the three domains are very much independent of each other, but still is a little bit of overlap. So, for instance, okay, comparing cell type. Archaeans and bacteria are both prokaryotic, okay, unicellular, no membrane-bound organelles. So there's still overlap there. Cell wall type is different across the board, so there's no sharing there. Membrane lipids are kind of a funny area because here the overlap is still maintained between eukaryotes and bacteria. Okay, they're phospholipid bilayers for both. Archaeans, for the most part, have their own distinct membrane type. Looking at the first amino acid in protein synthesis, so translation. So for archaeans and eukaryotes, it's both methionine. Bacteria, it's formylmethionine, so they're their own thing. And the list goes on. It's just to show you that, while there is still some overlap, the differences are pretty obvious between the three domains. Okay, another quick refresher slide in case it's been a while since you folks have had Gen Bio, looking at the taxonomic hierarchy. Okay, so, for a long, long time now, this has been in place. So, in the taxonomic hierarchy, we have different groupings or different ways of sorting out organisms. And the taxonomic hierarchy is the infamous domain, phylum, class, order, family, genus, species. So, as we classify organisms, okay, each organism is placed at each level. Okay, so humans have a domain, eukaryotes. We have a phylum, we have a class, we have an order, we have a family, we have a genus, we have a species. So all organisms have been assigned to each taxonomic level. Okay, so why are they least similar to most similar? Because as you work from the top of the taxonomic hierarchy down, there are more and more included groups. So for instance, there's 3 domains. Okay, but when you go down to phylum, there's hundreds of phyla. And then there's thousands of classes, and there's tens of thousands of orders, and there's more families, and more genera, and more species. Okay, so the 3 domains are the least similar to each other. Okay, archaea, bacteria, eukaryotes. That's as distinct as you can get. But as you add more and more groups, as you work down the taxonomic hierarchy, you start seeing greater similarity between the different groups. Okay, so there can be 2 closely related genera. Okay, they can be very, very similar. Not exactly the same, but more similar than different families. And families can be more similar than different orders, and so on. So you've probably seen this before in some biology class in the past. Okay, but the thing is, you related it to generally eukaryotic organisms. Okay, what I want to show you here is the fact that this does relate to archaeans and bacteria as well. Okay, they get assigned into these various taxonomic levels, and they have different names that you probably have never heard of before. So, for instance, we'll just look at E. coli on the end. Okay, we all know that E. coli is a bacteria, so you know the domain. Okay, but I bet you didn't know the phyla. E. coli is in the phyla Proteobacteria. It belongs into the class Gamma Proteobacteria. And within that class, it belongs to the order Enterobacterales. And with the family, this may be somewhat familiar, they're in family Enterobacteriaceae. And then we all know the genus, Escherichia, and the species coli. So, again, just looking at a bacterial example, okay, I just want to show you folks kind of how a taxonomic level works for a group of bacteria. Okay, so in family Enterobacteriaceae, okay, this is a taxonomic family, meaning you have a bunch of bacteria that are included in this family because they have similarities. And we've seen these in lab. So all of the enterics in Enterobacteriaceae are facultative anaerobes. They're all gram-negative. They're all bacilli. They can all ferment lactose. They're all motile. So to be in this family, they have to have all these characteristics in common. But look at what happens when you go down to the next taxonomic level. From family to genus. Okay, so common characters here. We got E. coli. Okay, tends to be totally harmless, a part of your gut microbiome, your gut flora. Compare it to, say, Shigella and Salmonella. Okay, these three organisms are in the same family. Shigella is a horrific pathogen which causes really nasty dysentery because it produces toxins. But then you have Salmonella. Salmonella is also a pathogen causing dysentery, but it does so in a very different way. It's because they have a capsule. It's not because of toxins. So, compare Shigella to Salmonella. They're in the same family. They both cause dysentery, but they do so in different ways. Get the Klebsiella. This is an opportunistic pathogen which can cause pneumonia in immunocompromised people. And then you got, say, Proteus species. Okay, Proteus species are another enteric, but they cause urinary tract infections. So, while they're all in the same family, as soon as you go on the next level of genus, you start seeing the differences between them. Okay, so let's move on from classification for now and start looking at methods of identification. Okay, so we're going to look at two overall schemes for identifying prokaryotes. First, we're going to touch on phenotypic identification. This tends to be more obvious stuff that you're familiar with, so identifying them based on behavior, or, say, biochemical characteristics. And then we're going to look at more modern techniques of genotypic identification, so looking at identifying organisms based on DNA or RNA sequences. Okay, so using phenotype, there's all sorts of methods, and these are more classical, but we're just going to hit on four of them. So microscopy, biochemical testing, serology, and then fatty acid analysis. Okay, so microscopy. This is not the best method of identification. Okay, microscopic identification is more situational. It has to be, say, a very specific pathogen in a place it doesn't belong. Okay, so for instance, in the greenish image here, you've got these kind of odd connected urn-shaped cells. Those are yeast cells. Okay, so imagine this is a vaginal sample taken from a patient that you suspect has a yeast infection. The vagina plays host to a number of bacteria, which is totally normal, but it should not play host to the yeast Candida albicans. Okay, so here we can identify it readily because it's in a place that doesn't belong. Now, in the bottom image, I feel bad if you ever have to deal with this, we have a patient where we suspect that they may have some sort of intestinal parasite. So the best way to figure out if you have an intestinal parasite is to dig through their poop. So you take a stool sample, you put a portion of it under the microscope, and you look for eggs. Okay, so we got a roundworm egg here. It does not belong in any healthy individual's feces. Okay, so again, readily identifiable because it's very situational. All right, two more kind of blunt examples. So identifying bacterial pathogens. And we're going to do so using our favorite differential stains, Gram stains. So in the top image here, say you have a patient with chronic pneumonia. Okay, you're not sure what kind of pneumonia. Okay, is it bacterial? Is it fungal? Is it viral? So you take a sputum sample, deep lung mucus sample, and you run a Gram stain. So folks, your lungs should be clean. There really shouldn't be any bacteria in your lungs. So, if you happen to see gram-positive streptococci, sorry, let me fix that. So if you happen to find gram-positive streptococci, that is Streptococcus pneumoniae all day long. Okay? Shouldn't have gram-positive streptococci in your lungs. Now in the bottom image, say we have a male patient with painful mucus discharge from his penis. Again, your urethra should, for the most part, be devoid of microbial life. It should be clean. So, you run a Gram stain. And most unfortunately, what do you find? Gram-negative diplococci. Okay? There's only one thing in the urethra that forms gram-negative diplococci, and that's Neisseria gonorrhea all day long. So again, these are very blunt methods of identification. Okay? Very situational. It's a good first step. Because what you'd follow that up with to confirm things is some form of biochemical testing. Okay, so, for one, you can start by looking at basic culture characteristics. So, you do a streak plate. You look at the colony morphology. Remember all the stuff we got from colony morphology? What's the elevation? What's the margin? What's the overall shape? What's the color? What's the consistency? Okay, colonies are often a lot like fingerprints for bacteria. But what's a little bit more reliable, and still done clinically to this day, is biochemical tests. Just like what we're doing in lab. So, the biochemical tests, here we're looking at all those different enzyme tests that we've done. Okay, so we utilize differential media to identify, what set of enzymes do they or do they not have. And these biochemical tests have classically been very reliable because no two bacteria have the same set of enzymes. Again, very much like a fingerprint. But the drawback to doing these biochemical tests, and say we're doing it the way we do it in lab, which is very classical, is it takes time. It's a lot of effort, it's a lot of setup, and it gets costly. Okay, so let me show you some different versions or different takes on biochemical tests. So for one, in lab, we've been doing a lot of biochemical tests. And when you do the unknowns, you're going to be doing a lot of biochemical tests. The thing is that you probably, in a real-world setting, wouldn't do all the biochemical tests. Okay, so for instance, you would set up or use what's called a dichotomous key. Kind of a flowchart or choose your own adventure of biochemical tests. So what we have here is a dichotomous key for identifying an enteric pathogen. Okay, so enterics are like we just saw. Members like E. coli, Shigella, Salmonella, amongst others. Okay, so you say run a Gram stain. Okay, you find out that you have a gram-negative enteric. So you have a good idea as to what sort of pathogen you have, so you don't have to run all the tests. So instead, you would only run a very specific set of tests. So for instance, you would start by testing to see if they could ferment lactose. Say they can't. So you go down the no route. Okay, next test you would do in this key is you'd run a citrate slant. Okay, so you run your citrate slant. If it's a no, so say that citrate slant stays green, then they have Shigella. Or on the other hand, maybe that citrate slant turns blue. Well, look at that. You identified they have a salmonella infection. Now go back to the top. Say that your enteric can ferment lactose. So you're going this way now. So again, you'd run citrate next. So you do your citrate slant. Okay, so if it's a no, then you want to see if they can ferment sucrose. Okay, so that sucrose tube turns pink, well then it's E. coli. But on the other hand, if that sucrose tube turns yellow, well then you've got a pathogenic strain of E. coli. So back up a bit. Okay, go back to that citrate slant. Say they did turn that citrate slant blue. So you run a VP test. So you see the acetone there? That's a VP test. So, if that VP test stays yellow, well, it's Citrobacter. If that VP test turns pink, well, then it's Enterobacter. Okay, so the point is that in a real-world setting, in a clinical setting, you'd have a rough idea of what sort of pathogen you're dealing with, and you'd only really have to run maybe 2 or 3 biochemical tests, not all 20. Another modern take on this is enterotubes. I've done enterotubes. We won't get to do enterotubes. It's a bummer. They are costly and not the most reliable things in the world. But an enterotube is a method of doing rapid biochemical testing. So for instance, in the top image here, you got this thing that looks like some sort of odd piece of candy. Well, each chamber is a different biochemical test, many of which we've already learned about. And the way this works is, you'll see that they're drawing a needle out of there. You take that needle, you touch it to a colony, and then you slip that needle back through all the chambers, inoculating them all at once. So, you inoculate all the chambers, you incubate, you come back within 24 hours, and then you check to see if there's any changes to those tubes. So, in the image we have the control tube versus the experimental tube. And you compare. Look for any changes in color. So, the way this works is kind of like a game. So you may see these little brackets of numbers here. There's a special scoring system you follow. Okay, so each chamber is worth a different amount of points. And you only count the points if the tube or chamber comes up positive. So for instance, they circled the number 2 here. Okay, that means that glucose was fermented. But they did not circle number 1. Okay, that means it did not make gas. So you only count the 2. So you work your way down the line. You work within each bracket, and you generate this code number. So we got 21007. That code number is everything in identification because they used to send you a binder. I imagine you can just look these up online. But if you look up 21007 for an enterotube, you'll find out that your organism is in fact Proteus mirabilis. So within 24 hours, you can identify an unknown organism very rapidly. So again, why didn't we do this? Well, for one, these things used to be like $5 a pop, and you generally have to do them in replicates of 3 to 5. So it adds up really fast. All right. So next up we have serological testing. Okay, so we talked about this with our infamous ELISA lab from a couple weeks ago, because in serological testing, we're trying to identify a pathogen based on either antibodies or antigens in blood serum. Okay, so again, folks, antigens are anything your immune system could recognize as foreign. And antigens are really any sort of macromolecule. Typically they're proteins, but they can be polysaccharides or lipids or nucleic acids. So in serological testing, we're looking for either antigens or antibodies because antibodies and antigens interact. They bind to each other. When you're sick with an infection, you make antibodies that bind to and target that pathogen, and only that pathogen. So when you're sick, you'll either have antigens from that pathogen in your bloodstream, or you'll have antibodies specific for that pathogen in your bloodstream. So all you have to do is identify one or the other. So that's what serological testing is all about. Okay, rapid detection of pathogens in blood serum. So that ELISA lab we did, for instance, we rapidly, I do air quotes that you folks can't see, rapidly identified HIV antigens from some mysterious person's blood serum. But another version of this is something you folks may be familiar with, rapid strep tests. So this is the old school rapid strep test. I'm not even sure if they do this anymore. But we have here is the slide agglutination test. So that's your fun word of the day, agglutination. Agglutination just means clumping. So when antibodies and antigens interact, they clump. So in the slide, we have two outcomes of the strep test. Okay, so the way this works is you swab the back of somebody's throat. You swab their tonsils because you want to see if you can swab up any of those strep cells. And then you apply that swab to a slide that contains antiserum. Okay, so antiserum is what that liquid is on that glass slide in the image. And antiserum just contains synthetic antibodies. So, if the individual has strep, you will see all these little white flakes, all these little clumps. Okay, that tells you that strep antigens were interacting with the synthetic antibodies in the antiserum, and you have clumping as a positive reaction. Now, on the other hand, in the negative test, okay, it just turns cloudy. So, for those of you folks who have had physiology, you've done something similar with blood typing. Okay, with blood typing, you're using 3 different types of antiserum to get your blood type. Anti-A, anti-B, and anti-RH. And in that test, you looked for clumping when you applied your blood to each of those antiserum. Because if you saw clumping, that would tell you your blood type. Okay, so the last and least common, it's so least common, I've never done it nor really seen it before, is fatty acid analysis. So here, this is a method of phenotypic identification based on identifying organisms on cellular fatty acid composition. So basically, what fats make up their cell. Because if you haven't really thought about it, fat composition is very much a personal thing. Very much a fingerprint sort of thing. So, even for bacteria, different species will differ in what sort of fatty acids they have. So the most obvious example, comparing gram-negative to gram-positive bacteria. Okay, gram-negative bacteria have that outer membrane made up of LPS. Okay, gram-positive bacteria have no LPS. So gram-positive bacteria are missing an entire type of fatty acid. But the thing is that FAME is far more refined than that. And the thing about FAME is that it's kind of boring. Because in order to determine what fats are in a cell, you have to perform what's called chromatography. So for those of you folks who aren't really hardcore into chemistry, there's all sorts of different types of chromatography. But typically, chromatography seeks to do the same thing. It helps to identify and differentiate different chemicals that are similar to each other. Okay, so I used to do carbohydrate chromatography. Okay, after feeding E. coli a bunch of sugars, I used to have to use chromatography to determine what sugars were left over. Okay, they're very similar to each other. This allows me to determine what sugars were left over in their growth medium. But this is where it's boring. This is basically what a chromatographic output looks like. A machine feeds you a series of peaks. So, those peaks, though, that you can see in the image are very important. Because when you run your sample through, these peaks show up at different times. And each peak represents a different fat. Okay, so for instance, this first peak here, that is palmitic acid, a type of fatty acid. The second peak is stearic acid. The third peak is oleic acid. The fourth one is linoleic. And the last one is linolenic. So let's say this organism had these 5 different fats in its cell. Okay, that can make it distinct. But on top of that, the different peak sizes matter as well. Okay, the bigger the peak, the more of the fat is present. Okay, and that's where things get really refined. Because while some bacteria may share the same fats, they won't have the same amount of each fat. Okay, so least common technique, but technically you can identify bacteria based on fat content. Okay, so moving on from using phenotype, what's become more tried and true and more common these days is identifying bacteria based on genotypic differences. So identifying bacteria based on their DNA or RNA, which is far more refined. And there's all sorts of methods to do this. Okay, so I just want to go over some of these methods very briefly. I don't want to get into the meat and potatoes of it because each one gets very detailed. But the first example of a genotypic method of identification is not the best, but it's in your book, so I'll talk about it. Identifying bacteria based on DNA base composition. Okay, so DNA base composition. What are we talking about here? So identifying bacteria based on how much A, T, C, and G they have. Okay, so really it's a ratio of GC content to AT. And we do this because of the base pairing rules, right? G and C pair up, A and T pair up. So when you look at a strand of DNA end to end, and you count up the number of GC pairs versus AT pairs, you start seeing some very interesting trends. So broadly, okay, organisms that have similar GC to AT content are fairly closely related versus those that have more distant or different GC to AT content would be more distantly related. So, for instance, let's do some comparing here. Okay, so let's start with E. coli. Okay, E. coli is sitting around 5149. Sorry, folks, that's percent. But then when you throw salmonella into the mix, we've got 52% to 48%. And then compare to Shigella. Okay, Shigella's sitting around, I think, 49, 51. I always forget the exact numbers. So they're a little different, okay, but they're still somewhat similar because all these guys are enterics. They're all in the same family. But again, the numbers aren't exactly perfect. Let me show you one that's really different. Let's talk about any of the Clostridia. Okay, this could be Clostridium difficile, Clostridium botulinum, tetani, whatever. Okay, Clostridia are way, way different because these guys have 30% GC and 70% AT. Okay, so you can see way different here. And then there's some organisms that are the opposite where they have like 70% GC and 30% AT. Okay, so not the best method of identification, but a fairly good starting point. Okay, so next up we have DNA fingerprinting. This one's kind of a funny one because I've only ever applied this to humans. And if you've had 110, Bio 110 here at Harper, you've already done a DNA fingerprinting experiment. Okay, so here's the way it works. In DNA fingerprinting, you take some unknown organism's chromosome. You remove that chromosome, and then you cut it up into a whole bunch of fragments using enzymes called restriction enzymes, which we'll talk about in Chapter 9. And these restriction enzymes set up a pool of DNA fragments. So you take those DNA fragments, you then visualize them using gel electrophoresis, which is again something else we'll talk about in Chapter 9. And what you get on that gel is what we call a DNA fingerprint, a unique pattern of DNA fragments. And this is something that's very species specific. So, in the image, we have a gel electrophoresis image. And I know I'm doing the chapters out of order because it's weird semester, but I will promise to talk about how these work. Okay, but each bright line you see is a fragment of DNA. And all this is a game of matching up the patterns of those DNA fragments. So, for instance, we can see that these two organisms, we'll call organism A, are the same. Their DNA fingerprints match up. The next two, we'll call organism B. Okay, those fragments match up. The DNA from those came from the same species. And then the last one here, we'll say organism C. Those match up. Okay, so every organism, every species, when you cut up its chromosome with these restriction enzymes, these little DNA scissor enzymes, they generate a very specific set of DNA fragments. And then all you have to do is visualize those DNA fragments and compare to identify your organism. So, if you've had 110 here, you know that this very experiment can be used with humans in order to identify murder suspects or parentage. All right. The third technique is something, again, we'll talk about more in Chapter 9, but using what's called PCR for identification. So, PCR is a method of making copies of a gene. Think of it as gene Xeroxing. But, in order for PCR to work, you need to create primers, so short sequences of DNA, that match up to a specific gene. So just to very simply show you folks how this works. Oops, that's not what I wanted. Alright, so say this is our gene. The way this works is that you're going to have two primers. Primer 1, primer 2. And these primers are just short sequences of DNA that you design. And then through PCR, you just copy that gene over and over and over again. So you make an exponential number of copies. We use this PCR method for identifying unknown organisms because these two primers can be organism specific. Because PCR is all or nothing. Either those primers will bind to some target gene, giving you DNA, or nothing happens. So, using PCR for identification, a nucleic acid amplification test, you utilize a series of organism-specific primers. Okay. If any of the reactions work, so say you run 20 different reactions to identify 20 different pathogens. If any one of those reactions work, if any single one gives you amplified DNA, that means you've identified your pathogen. Okay, this is a very sensitive reaction. Okay, it works with very small numbers of bacteria. But the big limitation here is you have to have these primers. These are something that you design. So you have to have a rough idea as to what you're looking for. But PCR is something more we'll talk about in Chapter 9. Now, the fourth method is, honestly, is what most folks use for identifying based on genotype. And it's something we talked about briefly before with Carl Woese. Sequencing genes for rRNA. Okay, what we call ribotyping. So the idea here is that all organisms have ribosomes. Therefore, we all have genes for rRNA. And we like to target those genes because there's very little genetic variation. Very little mutation happens in those genes for rRNA. So that means related species of organisms will have very similar rRNA sequences versus more distantly related organisms where you can have more mutations that have accumulated, so more differences. So in ribotyping, all we're trying to do is to get an organism's rRNA gene sequence. And once we get that sequence, because there are massive catalogs of rRNA sequences already out there on the internet, you just get your sequence, blast it, literally what it's called, blast it on the internet, and look to see if you get any hits. Okay, so ribotyping is where we use rRNA gene sequences to identify an unknown organism. Alright, so this last method is unfortunately another thing we'll talk about a bit in Chapter 9. Nucleic acid hybridization tests, aka using DNA probes. So, probes are a lot like those primers in PCR. They're something you design, and in this experiment, we utilize a whole bunch of different species-specific probes. So you have a whole library of probes. And when you design these little short sequences of DNA, these little DNA RNA probes, we also tag them. In the past, we used radioactive isotopes. These days, though, we use a little bit more friendly fluorescent dyes. So, for instance, in the diagram, you design your tagged probes, and then you take DNA from an unknown organism. Okay, the strands are denatured, meaning that they're separated. And then you mix. So mix together the DNA from the unknown organism with the probes. Now, these probes are all or nothing. Because again, you design these probes to bind to DNA different pathogens. So, in the top here, if it doesn't bind, okay, that tells you that those probes don't match that organism. But on the other hand, if the probes do bind, that tells you have identified your unknown organism. And you would do this in a whole bunch of different test tubes, and you would look to see after if any of them glowed under fluorescent light. So this one, for instance, this one would glow under UV light. So you take a library of probes. You set up a bunch of different reactions. You take DNA from an unknown organism. You expose them to these probes. If the probes bind to the DNA of this organism, whichever probes bind, that tells you what your unknown organism is. Okay, so reinforcing this stuff, we have a little bit of a wrinkle we have to talk about. Strains. So, strains are something that's common to bacteria. You don't really see it with eukaryotes. So, in the taxonomic hierarchy, we saw that it ended with species, right? It's domain, phylum, class, order, family, genus, species, and then that's the end of the road. And that's nothing lower than species. Well, with prokaryotes, technically we're going to break the rules a little bit. Because you encounter strains, a term I'm sure you've used, but you may not realize what it means. A strain is a genetic variant or subtype of some specific species of bacteria or, say, archaeans. So, let's look at the examples to make sense of this. So, being able to differentiate between species is really, or different strains is really important. So, for instance, in the first example, E. coli B versus E. coli O157H7. Okay, E. coli B, the B is the strain designation. Okay, O157H7 is that strain designation. So they're both E. coli. But what's the difference? Well, E. coli B is basically wild type. O157H7, on the other hand, is the infamous Jack-in-the-Box strain, identified in the 1990s. O157H7 is very much pathogenic. It can cause enterohemorrhagic fever and very, very bad dysentery. So, what's the difference? Okay, they're both E. coli, but O157H7 is pathogenic because it has a gene for the Shiga toxin. Okay, that gene doesn't make it a new species. It's a small genetic difference which makes it a different strain. The other example, Staph aureus versus MRSA. Okay, we've been working with Staph aureus in lab all semester. For the most part, totally harmless. But then you got MRSA, which is methicillin resistant Staph aureus. So, one is sensitive to most antibiotics. The other is extremely resistant to a great deal of antibiotics. So they're both Staph aureus. So what's the difference? MRSA has one extra gene. A gene that makes it resistant to an entire class of antibiotics. So, that doesn't make it a new species, it just makes it a strain. But the point is, within a species, especially with bacteria, there can be tons of strains. And in many cases, due to human health, it's very important to identify and differentiate these different strains. So, how do you do that? Well, there's a lot of redundancy here. you can utilize a lot of the techniques we've already talked about. So, biochemical typing. So, before I go further here, when choosing which method you want to use to identify your strain, keep in mind these are very situational. Okay, it's a right time and a right place for each of these. So, biochemical typing, for instance. If there's some sort of biochemical difference between the strains that you can identify, you can use this. So, for instance, identifying different strains based on hemolysis. So, utilizing blood agar. Is it beta hemolytic or is it alpha hemolytic? Serological typing. This one's been pretty common, especially with Salmonella species. So here, we want to identify the strains by looking for differences in surface antigens. Okay, so what are those little chemical differences on the surface of the pathogen? So like I said, Salmonella species are notorious for this. There are probably more strains of Salmonella out there than any other group of bacteria. And the only good way of telling many of these strains apart is through serological testing. So for Salmonella species, we rely so heavily on serological typing that we don't actually call them strains for Salmonella. We call them serotypes or serovars. Phage typing can be used. Now, this is the one we didn't talk about. Phage typing is utilizing viruses, specifically bacteriophages, to identify different strains. When we get to chapter 13, we'll talk about this a bit more, but viruses are really, really picky about who or what they'll infect, down to even strain differences. Okay, so we want to look for strain or host-specific bacteriophages. So here's a phage typing experiment. So first you start by inoculating your broth, or inoculating your plate, and then you add these little droplets. Each droplet is a different virus. So you add your different viruses, you incubate, you come back the next day, and you look for these clearings. Okay, those clearings are what we call plaques, which is dead cells killed by the virus. So each plaque tells you which virus the bacteria was sensitive to. Okay, because there's no plaques, the virus didn't do anything to it. So why do we care here? Well, compare the two plates. Okay, you have two very different plaque patterns. Okay, so say these are both different strains of Staph aureus. Okay, the different plaque patterns can be used in identifying and differentiating the strains. Fourth example, genomic typing. This is very common in identifying different strains these days because, again, strains are different because of small, subtle genetic differences. So just look for those little genetic differences. So pick your favorite genomic method. Okay, here in the example, I use DNA probes. Okay, so identifying wild type E. coli versus that nasty O157H7. Okay, you can use probes that will only bind to the gene for the Shiga toxin present in the pathogenic strain. Okay, so the last method of characterizing strain differences is antibiograms. Now, this is one we haven't done yet, but we will get to do in lab later this semester. So in antibiograms, and again, very situational, we're trying to identify and distinguish between strains based on antibiotic susceptibility, meaning what antibiotics is each strain sensitive to. So let's just jump to the experiment here. So we've got 2 plates. Say we've got normal Staph aureus versus MRSA. Okay, each plate's got a bunch of colorful discs. Those colorful discs have been soaked in different antibiotics. So each color is a different antibiotic. So what you see around the disc, that's what we call a zone of inhibition. The idea here is the bigger the clearing, the more effective that antibiotic is. Okay, small clearings, not effective. So the point is, compare the 2 plates. Okay, normal Staph aureus on the left is very sensitive to a great deal of antibiotics, versus MRSA, which is resistant to a far more antibiotics. So right there you can see a difference in the size of the zones of inhibition. Okay, so here you can roughly identify Staph aureus versus MRSA. Okay, so the last thing we're going to talk about here, and we're almost through this, is kind of putting this all together, coming back to classification after touching on what we've learned about with identification. So, phylogenetic classification, so again, deciding who is related to who, was classically, historically based on phenotype. Okay, looking at biochemical tests, looking at culture characteristics, how they grew on plates or slants. And that was great for a long time, right? They look the same, they behave the same, therefore they're related together. But as recent history has shown, using phenotype is not particularly reliable. Okay, because you can have organisms that look and behave the same, but when you start digging into their DNA, you find out there's more genetic differences to be found. Okay, so there's a great deal of, say, smaller mutations that have accumulated, making organisms more distantly related than one would think. So, as we've mentioned, today, modern techniques rely on identifying prokaryotes based on genotype. Okay, it's proven to be far more reliable for the last 30 years. So, what aspect of genotype are we looking at here? What regarding an organism's DNA or RNA are we focusing on? Well, as we talked about with Carl Woese, the most common method is utilizing 16S rRNA gene sequences. So, something I failed to mention here, folks may wonder, what's up with 16S? Right, we've talked about 30S, 50S, maybe 70S. What the hell is 16S? Well, the thing is that ribosomal subunits continue to break down. Okay, so every bacterial species out there has a gene for 16S rRNA. Okay, it's a very small gene. It sees very little mutation. It's highly conserved. So it's been the preferred gene sequence for classification of bacteria. So as we sequence 16S rRNA gene sequences and we start comparing them, what we're trying to construct is what's called a cladogram, a phylogenetic family tree. So let's bounce back and forth here. So, here's an example of how to build a cladogram, and this is very crude. So see, what we have here is a portion of 16S rRNA sequences with 4 different lactobacillus species. That looks terrible. And we want to see, of these 4, who's the most closely related, who's the most distantly related. So you take these 4 16S sequences and you start comparing. You want to figure out similarity. So, go down, you can see how similar or different they are. So, we've got, say, Lactobacillus brevis compared to Lactobacillus sanfrancensis. they only have a 50% similarity. But then when you compare Brevis to Acidophilus, look at that, they're 70% similar. And then you just keep working your way down. So Brevis plantarum is 60%, Sanfrancensis Acidophilus 50%, and so on. So you just mix and match all of them to figure out initially percent similarity. And then once you've got that percent similarity figured out, then you can start drawing up your cladogram, your family tree. Okay, so let's start with our last common ancestor between these four. Okay, the organism which gave rise to all four of these Lactobacillus species. So we put Sanfrancensis out on its own because in every case it was the least similar, only 50% similar. So that's the most distantly related one. So then we work up our way to the next branch point, those that were 60% similar. Okay, so that brings us to Plantarum. Okay, it's the second most distantly related one. And that brings us to our next branch point. We have Brevis and Acidophilus, which had 70% similarity. Those two are the most similar, and they probably shared the most common ancestor between the lot. So this is a very crude cladogram. Okay, you just build these family trees showing where these little divergence points happened, so where mutations accumulated in our RNA gene sequences. So looking at more realistic examples here. Okay, what you have here is an actual 16S rRNA gene sequence alignment. So we got 20 different organisms, and we're comparing all 20 16S rRNA sequences. So you can see how similar those sequences are, right? Those color bars line up pretty well until you get about 2/3 of the way through. And then you start seeing the differences. The point is, no two organisms should be the same. They should all be distinct. So what we have here is kind of a more realistic cladogram. Okay, the one we saw was very crude. This is one that's been expanded upon a little bit, but the idea is the same. You organize, classify these organisms based on similarity. And once you do that, you can work backwards and see where those little evolutionary divergence points happened. Okay, and this was a very small one. Okay, a real cladogram, including all organisms, is absolutely massive because it includes millions upon millions of species. So the last thing I want to leave you folks here with something to think about is the fact that we're quickly moving on from using 16S rRNA gene sequences. And these days, we're starting to rely on whole genome sequencing. So sequencing an entire chromosome in a bacterium and then cataloging it. So we're generating these massive catalogs of entire genome sequences to paint a very different phylogenetic picture. And what we've rapidly discovered through doing this is the fact that horizontal gene transfer is more common than we ever thought, especially amongst prokaryotes. They have swapped DNA back and forth down to the level of strain. So, creating phylogenetic trees is becoming very difficult. So those nice, clean branch points that all ended with an individual species, problem is that may actually be impossible. Maybe more like a phylogenetic shrub. Because of horizontal gene transfer, these branches have become impossibly interwoven. So we may have to redefine the taxonomic hierarchy just because of how much DNA gets swapped on a day in, day out basis. Gonna have to redefine what it means to be a species. Okay, so that's it for chapter 10. Again, I'm going to try to find time later this week to review it. so that I can field any questions and just hit on some important points. Thank you.